All right, so we're gonna look at the respiratory system very briefly. Not a whole lot of difficult stuff in the for the respiratory system for lab at least. What you're looking at here, of course, is the spirogram. Over here, this would be the tidal volume, abbreviated TV. This is what you're doing now, slowly breathing in and out. Right, and this IRV stands for the inspiratory reserve volume. So that's how much air you could potentially breathe in above what your normal breathing is. Then we have the inspiratory capacity. What we've done is added these two together. So it tells us a function of the lungs, the total amount of air you're able to breathe in. All right, and we get down to looking at the other side of that then, the expiratory reserve volume, how much space you have left to be able to breathe out. So this is potentially a look at the amount of air that's still left in the lungs after a full expiration. And then with a full deep breath, we see the RV down there, the residual volume. This is how much air is left after fully expiring or forcing out all of the air in your lungs. So if we put these two together, and what we're left with is the functional residual capacity. So again, looking at the overall health of the lungs, how much you're able to blow out. Now, when you go see your pulmonologist, if you have one, but you completely breathe in and then completely breathe all the way out, that is considered to be the vital capacity, how much work your lungs are actually able to do through an entire breathing cycle. And the TLC down here doesn't mean what you think it does. That's total lung capacity. So how much your lungs could potentially do in the terms of work. Traditionally, we've asked simple questions such as to calculate one of these. Uh, Usually what I'll do is ask you to calculate the inspiratory capacity, and all you do is add these two numbers together. I haven't asked that in a while. It could pop up. If it does, it will be that easy, though, just to make sure you understand kind of how that works. These next pages here are kind of explaining everything that I just told you. Nothing really questionable there. You're probably going to have this table with some of these values removed, and I'll ask you to identify what this area was to represent. So get to know what these abbreviations mean, and you can abbreviate on the lab exam. I don't have a problem with that. So if you just want to memorize where these abbreviations go on this table, I might have one of these things that's a hotspot where I just have this entire table of no letters and just ask you to touch or click on inspiratory reserve. Then you just have to click in this area here to get credit for that. So it'll be something simple like that, hopefully. All right, again, moving on, not too much from here. Just gives you some calculations. But like I said, if I were to ask one, it would be probably this one here. That would be the easiest one. So make sure you know how to add, at least for this part, in case I do ask that one. All right, so moving on from that, we're going to see pictures from the pig, or may use a model, so the external nares or nares, however you want to pronounce it. Those are just the openings for the nose there. This one is on one of the practice quizzes, I believe. It's not a trick question. If I ask you for the structure inside the mouth there, that's the tongue. I'm not sure exactly what two is supposed to be pointing at there. Oh, well. It's probably something else I forgot to put on there. Now, I've seen these before in anatomy too, with your superior, middle, and inferior nasal conchae, also called the turbinates. Either way, but let's see, get past that. This is how you'll be tested on this. You have the half head model. So each one of the raised ridges is the conchae or conchae. The ridges in between, those are the superior, middle, inferior nasal meatus. All right, so you'll remember nasopharynx, oropharynx, laryngopharynx down in this area. The structure back here, the uvula. And then these ridges here. The top one, know that that is the superior vestibular fold or the false vocal cord. Either one's okay. But I do want you to know that the true vocal cord is inferior to the false vocal cord. So the inferior vestibular fold is the true vocal cord. I don't know if there's another. Yeah, I've already mentioned that part, so we're okay there. And there's that question. That's exactly how this will be a question on the test as well. Use these brackets, and probably I'll just take this exact picture and remove the words and ask you to identify one of those regions. And it will say identify the region. All right. I had that. Oh, well, I was looking forward to try to see. I'm missing a slide here, it looks like. But you may be asked for this area in here. What bone makes up the hard palate? Just make it as simple as possible. It's palatine bone and the palatine process. And if I ask you what makes up the soft palate, it's all skeletal muscle. That may appear here in somewhere in a little while, but I didn't want to keep going and then forget to talk about it. All right, so this picture is kind of representative of this model that we have in the lab. So you'll need to know that this prominence here, this is the thyroid cartilage, and it is made of hyaline cartilage. Read the question closely on the exam. I'll say identify the structure, thyroid cartilage, and then identify the tissue that makes up this structure, hyaline cartilage. The epiglottis is kind of hiding back in there. I'll get a better picture of that. Or yeah, I'd probably have to get a better picture. I don't see a good one on there. But the structure is epiglottis. And actually, let's look at this again back here because I've used this before. This blue thing, all the blue on here is supposed to represent cartilage. So glottis is just an opening, it's a hole. So the epiglottis, remember epi means on top over covering. So when you swallow, this flap comes down the cover of the glottis so you don't inhale your food and drink. So that's the epiglottis is the structure. The tissue that makes it up is elastic cartilage. Remember that, very important. Boards love those questions. So, hyaline cartilage for thyroid, here we go. Hyaline cartilage for thyroid cartilage, elastic cartilage for the epiglottis. All right, a little soft spot between there, and like I said, I'm going to use this model, so let's look at it the right way. So this feature in here, cricothyroid ligament, and then this specific feature, I'll ask you to identify this specific feature. That is the cricoid cartilage, right? The first ring of cartilage at the top of the trachea, and this structure down here is the trachea. Probably won't ask you for the hyoid bone. We identified that in Anatomy 1. There's no reason to go back over that again. I think you understand what it is. All right, let's see here. Yeah, you do need to know ciliated pseudostratified columnar epithelium in the trachea, and know that there are goblet cells there. The goblet cells make mucus, of course. You'll need to know the carina, and that is kind of a feature type thing. This is a silly picture here. That's the inside look, so we're looking down from the top of the trachea here. Looks like probably right and left primary bronchi. The carina is the middle portion there. So if we look here, that little pair of underwear, that's the carina. And I don't remember if I put on the test structure or feature. It seems to me that would be a feature. But do get to know the right and left main bronchus or right and left primary bronchus. Get to know those secondary bronchi. Remember your rights and lefts on this test. It is important. This whole semester, rights and lefts are important. On the previous exam, I went back through your quizzes and exams, and I believe I gave partial credit if you didn't put right or left. So be sure you put your rights and lefts. But the primary or main are the first two branches. The next one is secondary bronchi, right and left, and then tertiary way out here. There was an extra credit question. I don't know if I may put it on here in case you want extra credit. But any foreign object, if you were in my lecture, you heard this story of a little boy that was shoving peanuts up his nose, and one of them accidentally went down through the nasopharynx, oropharynx, and laryngopharynx, and turned and went through the trachea. So any foreign objects that enter the trachea, just pulled by gravity, usually end up in the right primary bronchus or right main bronchus. So this is usually where we find any foreign object. I've asked that before as an extra credit question. I may do it again this semester because y'all love extra credit. It's kind of fun. All right, moving on from there. And yet another picture of some model. Very good. I don't even know if this is, I don't know. I found it online and thought you might like it, so I put it on here. Ooh, I do need to talk about that though. Be sure you know that the alveoli are the actual sites of the gas exchange. That will be a question on the exam. Be sure you know that's where that happens, not the alveolar tree or any of the bronchioles or bronchi. The alveoli is where the actual gas exchange occurs. You need to know about the lobes, and I'll have probably maybe this picture, probably the actual picture of the model. You'll need to know on the right side, there's 3 lobes. On the left, there's 2 lobes. You need to know that the reason for that in most people is because the heart is on the left side. So crowding out the lungs there. So very easy to identify these. The right superior lobe, right middle lobe, right inferior lobe. Over here, the left superior, left inferior. And this feature, and what we call a feature, cardiac notch. All right, what else do we have? You need to know that the diaphragm is the primary quiet breathing muscle, just below the lungs there. Here again, we see this model, so you can get used to seeing how I would question that. The feature there, the cardiac notch, diaphragm down below. All right, that is hyaline cartilage. If I ask this, it'd be because I don't have any other questions. And my daughter's playing with dolls in the background. If you hear a bunch of shouting, she plays these fun scenarios with her dolls, and they do a lot of screaming. All right, so we've talked about this. There's a very disgusting look once again. Probably want to ask this question this way. It's just an interesting angle. That again is elastic cartilage. And, don't plan on showing a whole lot of tissue pictures, but if I run out of questions to ask, I will do this. So throw back to Anatomy 1. All right, this is not gonna make as much sense. I won't ask a question like this. On the pig, there's more lobes than we have. So you won't be asked that way. Like I said, we're not gonna be pigologists. So know the normal human anatomy as far as the lobes of the lungs go. All right, this goes a little too deep too, so I'm not gonna question too much from there. There it is again, your ciliated pseudostratified columnar epithelium. Come on, your goblet cells. All right, continue to move on. Simple squamous epithelium in the alveoli. So I could show you this picture and ask you to identify the tissue, identify the location. So the alveoli or alveolus, either one's okay. All right, now this is something that you should already know, but they wanted to make sure that we reinforce this. I guess I've never really understood why we have to teach this. But for this part, these will be fill in the blank questions. So you'll see these exact definitions, I guess you want to call them, or terms, and then you'll have to fill in this group over here. So most of these should make sense to you. Your hypothesis, your educated guess. The control group is the group that's been given placebo usually. The experimental group gets the actual drug or substance that's being studied. I gave little Tylenol examples here to make sure you understand it. Appropriate conclusions, that's just stating what you found, sample size. This is one of the most important parts of a study. You need to have an appropriate sample size. So the more people you have or the more subjects you have in the sample or in the study, the more reliable the outcome will be. And then your experimental errors, people that don't know how to do math, those kinds of things, anyone that leaves an experiment. I can give you a little story with this, I guess, as long as my battery doesn't die. You might have to plug in a second. But C. diff, if you've never heard of this, Clostridium difficile, it is a bacteria that's normally in our gut, and it's kept in control by all of the other bacteria. If you take large dose of antibiotics, you have long-standing diarrhea, you wash out most of your good bacteria, or kill most of your good bacteria, and Clostridium difficile stays there. If it starts to grow out of control, like we see in immunocompromised patients, for instance, then it becomes very nasty. And in medicine, you're going to have to deal with these C. diff patients. That means you have to robe up like you're going to see someone with a coronavirus. It's very highly transmittable, and it's nasty. Large volume, usually green or blue-green diarrhea, very large volume. A lot of these people will die of dehydration or electrolyte imbalances. So what we had is a physician in a hospital in San Francisco had an idea of how to combat C. diff in his patients that were getting it over and over every year. So he got a small group. I think it was 8 total people in his group. And what he was doing was saying, anytime you have to go to the restroom, go. You'll stay here for your study all day, and then we'll send you home, do all kinds of doctor's visits, that kinds of thing. And then what we're going to do is implant you with normal human poop. And basically they would go up through the rectum to implant that stool. And, a lot of people are uncomfortable with that. Some people left, but he kept his number 8. I don't know why the magic number was 8. So he had everyone in agreement, and what he's going to do is install this stool to find out if it cured C. diff better than the harsh drugs that we use. And everyone except for one person went through with this. This one person decided he didn't want someone else's poop installed in him, but he didn't tell the group that he didn't want to do it. So what he did at the end of the day was he went home and gave his roommate a bag and said, hey, when you have to go, could you put it in this? And I know this is a lovely story. Hopefully you're not eating during this. But for some reason, he trusted his roommate more. So he personally himself installed his roommate's sample, I guess if you want to put it that way. The thing was that the cure rate was 100%. Every single person was cured within a day of that installation. But that would be considered an experimental error because he did tell them later what he had done. So his result had to be thrown out. A larger study was done, and it was found to be 98% cure rate. So that's kind of how we fix C. diff. Now we'll give a little drugs, antibiotic that's specific for C. diff, and then you'll have this implant, or either now there's a poop pill that you can take orally that's supposed to help out. So a little example for you there to make sure you understand how all this works. But like I said, your exam questions for this can be very easy. I'll give you these words, you fill in that word. All right, you need to know a little bit about the enzymes for this test as well. Know that the enzymes help speed up reactions, and that's why we're able to function normally. If we didn't have enzymes, then the chemical processes that keep us alive would be very slow. So slow, in fact, that we'd spend our days lying around waiting for a meal to pass by so we could grab it. So know that if you increase the concentration of enzymes, you increase the rate of reaction. And going the other way, if you decrease the concentration of enzymes, you decrease the reaction. These are concerns with people that are getting very sick. They're denaturing their enzymes. and they're not able to push these reactions along. Could eventually lead to death or organ failure. The pH and temperature act differently from the concentration, but they act similar to each other. So when it comes to pH and temperature, you need an optimal range to make the enzymes work. So lucky for us, the perfect pH for the enzymes that keep us alive and healthy happens to be somewhere between 7.35 and 7.45. So if that doesn't sound familiar to you, then you weren't listening early in the semester or last semester when we said the normal pH for the body is 7.35 to 7.45. Also, the temperature range for the uh, enzymes to work normally happens to be the range of the normal human body temperature. So if we upset our pH balance, or if we start to get a fever that gets too high and lasts for too long, or if we're out in the cold for too long, then you're going to denature the enzymes. And that's going to lead to your chemical processes slowing down and eventually leading to organ failure and death. You do need to know that term denature, though. So if I ask on the exam, uh, I can't remember exactly how I worded it, something along the lines of what we call the process of enzymes being broken down or deactivated. That's denature. All right, so hopefully this is helping you out and you're staying healthy. So we'll be on to the next section pretty soon. I'll start uploading the quiz. I don't remember when the calendar said it was supposed to open, but either I'll look back at the calendar and either open it today or next week, and it'll probably be open for a couple of weeks to help you out.